Okay, welcome everyone. I think we can get started. It's my pleasure to introduce my colleague, Georg Wohlfahrt, who is a professor and head of the Department of Ecology at the University of Innsbruck. He's also chief editor of the journal Agricultural and Forest Meteorology. So Georg's work is in the area of biometeorology and land-atmosphere interactions. He spent a lot of time thinking about partitioning fluxes into their components, so with respect to net ecosystem CO2 exchange. So today, Georg is going to be talking to us a little bit about some of his carbonyl sulfide flux measurement as a way of partitioning. He's also done a lot of work using sun-induced or solar-induced fluorescence to pursue some of this partitioning. So he's at the forefront of some of these advanced partitioning methods. I look forward to seeing this seminar on gross primary productivity, quantifying the unmeasurable using flux measurements of carbonyl sulfide. I'll turn it over to you, Georg.
Thanks very much for the introduction, Tim. And good afternoon, everybody. So it's a pleasure to talk today about my sort of favorite trace gas, and that's carbonyl sulfide. So the talk will be structured like this. We start off with a definition of gross primary productivity, because actually that's then important in the following, so that we're all on the same page. Then I will dwell a bit on the theory which is behind, so the idea to use carbonyl sulfide, and I'll abbreviate it as COS. Some people like to say OCS as a proxy for GPP, and then I will present some recent developments, some complications associated with COS, and finally wrap up with the conclusions. So we start off with defining the gross primary productivity, and here I need to warn you, in the following you'll see a few equations, but that's really required in order to fully understand what this is about. So on the leaf scale, we can use the following definition of gross primary productivity, because and this emerges from the various terms that contribute to the term on the left side here, and that's the net photosynthesis. On one hand, we have this term here, VC, which is the carboxylation rate. Some people have called it the gross photosynthesis or the true photosynthesis. So this is really the initial step at which the enzyme Rubisco fixes CO2. But whenever there's oxygen at the same time, and inevitably, CO2 is lost again, and that's due to the oxygenation. And subtracting this oxygenation from the carboxylation, we actually get what has in the past been called the apparent photosynthesis, and what we sort of define as the gross primary productivity. And then we would still have another term, and that is the net photosynthesis, and this one then includes emissions of CO2, which are due to mitochondrial respiration that continues in the light. So this is something that Longhang Gu and I came up a few years ago. Graphically, this can be imagined like this. So this would be a kind of a light response curve that you would do, for example, with a leaf gas exchange parameter. In this case, it's model derived. So we have the CO2 flux on the y-axis, the incident photosynthetically active radiation on the x-axis. And the uppermost curve here is the carboxylation. And at the same time that carboxylation occurs, we lose CO2 because of the oxygenation. So this is down here, shown as a negative numbers, and that brings us down to this pink line here, which is the gross primary productivity. And then the net photosynthesis is a bit less, and this is due to and that's shown in green here, due to the mitochondrial respiration that continues in the light. So this is the leaf scale. At the ecosystem scale, in principle, we have the same story. However, it's a bit more complicated because in addition to the leaf respiration shown in green here, we have other sources of respiration, notably from below ground components. So that would be, for example, root respiration. So that would be autotrophic respiration, but also very much heterotrophic respiration. But in principle, we again can define GPP in the same way here, and here it would be this light blue curve. Yes, and GPP is really a key term. What's shown here is this is a sketch of the carbon cycle, and you can see GPP here is at the center. So this is the kind of primary step at which carbon is assimilated. Some of it is sort of lost relatively soon or immediately. The rest is used to build up the biomass, for example, in the forest, the woody biomass. And eventually, at some point, either these above ground or also below ground parts, they die off, they fall on the ground, and they are then decomposed, and then this CO2 is in this way returned back to the atmosphere. Maybe the wood is harvested and turned into wood products, or maybe there's a fire or something like that. And really GPP is here at the heart of all this. And moreover, GPP, because the CO2 needs to fuse into the leaf through the stomates, also links to the water cycle via the stomatal conductance, and because transpiration is a major pathway to get rid of energy, it also links to the energy cycle. So for all that, GPP is sort of really a key component, not only for the carbon cycle bit, but because of the links to the water and the energy cycle as well. Unfortunately, at least at the ecosystem scale, we can't measure GPP directly. At the leaf scale, we can play some tricks, and then we can do it, but not at the ecosystem scale. So we need to apply indirect methods to actually estimate GPP via what I call some proxy variables. And thus it's not a direct measurement, but it's an indirect approach, and we're using this proxy and some modeling framework in order to estimate then the GPP. So this is, for me, it's really an important point, and I want to illustrate it with an approach that many of you may know. That's the CO2 flux partitioning. That's, for example, applied in a consistent fashion within the FluxNet project. So whenever you download some FluxNet data from some site, the GPP and the ecosystem respiration have been derived from such a flux partitioning algorithm. And here it's important to keep in mind that we have some measurements, they are fed into a model, and then we get the GPP. So in case of, for example, the nighttime flux partitioning, the measurements are the net ecosystem production, or the net ecosystem CO2 exchange, if you will. And then we have a model. And for the nighttime approach, this model makes two major assumptions. It assumes, number one, that what we measure in terms of CO2 exchange in the night is equal to the ecosystem respiration. And then, and that's really a major assumption, that the ecosystem respiration in the night equals the ecosystem respiration during the day and is a function of temperature. And what we then do is we simulate this ecosystem respiration during the day as a function of temperature. We put it then into this equation here. We rearrange the equation, and then we get GPP. So that is what I mean with that we're using proxies in order to, or we have to use proxies in order to estimate GPP, because we cannot measure it directly at the ecosystem scale. So what kind of proxies do we have? And here, conceptually, I find it useful to separate sort of the the entire process, starting off with the light reactions, where the energy and the reduction equivalents are produced in order to then fix the CO2. So eventually some carbohydrates are generated by photosynthesis, and eventually those are then at some point respired and the CO2 is released. So for this first part, we have basically two methods. One is one that Tim already mentioned, the sun-induced or solar-induced chlorophyll fluorescence, and there's another one based on like a reflectance index, so-called photochemical reflectance index. And both of them, both of these approaches, they target sort of the light reactions. Then we have carbonyl sulfide, and as you'll see in a few minutes, carbonyl sulfide rather ties into the diffusion part of the photosynthesis, so the diffusion of CO2 into the leaf via the stomata. And then we have a bunch of methods that are placed at the intersection between the uptake of CO2 and the release, so the CO2 flux partitioning as it is applied in the FluxNet. Processing of data falls in this category. Then we can do flux partitioning using stable isotopes of, for example, CO2. And then there's one very peculiar method, that's flux variance partitioning, that also uses the water vapor flux. So this method actually uses high frequency measurements of CO2 and water vapor for the partitioning. And since all those are proxies, we'll sort of never really know the truth. And so in my imagination, like we as scientists using different methods are like the blind man and the elephant. So the blind man encounter this elephant, and everyone gets hold of a different piece of the elephant, and according to this piece, everyone develops a different idea so what they have encountered, and so the different methods they most likely will give different answers. And actually, what I think is the way forward is to combine more of those methods in order to apply a multiple constraints approach on GPP. Okay, so this was kind of a bit of a background, and now we're turning to carbonyl sulfide, and I want to explain the idea of using it as a proxy for GPP using this slide here. It shows the cross section of the leaf. So here would be the upper side and the lower side down here, and here would be a stoma. And what's shown here is the pathway of CO2 on one hand and COS on the other hand into the leaf. And so we have here points of concentration, and in between we have resistances that those two molecules encounter on their way. So for CO2, it needs to diffuse through the boundary layer, through the stomata, then through the mesophyll, and eventually it then gets carboxylized by the enzyme, by Rubisco. COS has pretty much the same pathway, but the endpoint is not the Rubisco. It's a bit further upstream, somewhere in the cytosol, where the COS is catalyzed by an enzyme called carbonic anhydrase. So the first thing to take away from this slide is that the diffusion pathways into the leaf are not identical, but they're very, very similar. So the second point that is important here is CO2 diffuses into the leaf in this way, but at the same time, and that's something we've seen in the first equation, there's an emission term here as well. So that's the daytime respiration. So at the same time that CO2 diffuses into the leaf, there's an outward flux due to mitochondrial respiration. While COS, it's a one-way flux. So it just goes into the leaf, and the concentration at the endpoint is basically zero, because the enzyme is very efficient in catalyzing COS. So there's no outward flux, so no respiration like COS flux. And taking those two things together, it means that we have this tracer, COS, whose pathway is almost identical to the gross primary pathway, but there's no emission term. And that's sort of in a simpler way, that's the idea behind using COS as a tracer or as a proxy for GPP. So the idea is if we can measure the uptake of COS by a leaf or by a plant canopy, that in this way we can estimate the gross uptake of CO2, so the gross primary productivity. Yeah, so you may have been wondering, so where does COS, where does it come from? What's the ambient concentration? So the ambient concentration of COS is roughly, so averaged over a whole year and over the entire globe is roughly 500 ppt, so parts per trillion. So almost by a factor 10 to the power of 6, smaller than CO2. And there's more differences to CO2 as well. So really the big source of COS is the ocean. So COS is produced either directly from organic molecules in the surface water of the ocean, or precursors, CS2 and DMS, so dimethyl sulfide, is produced in the ocean and is then oxygenated to COS. And those two together are the big sources of COS. There's a biomass burning source of COS, and then there's also an anthropogenic source of COS, or the precursor substances. For example, the rayon production is a major anthropogenic source, but those two together are by far not as big as the ocean source. Then there's soils which play a role, and soils may either be a source or a sink. So this is something we'll come back to later. And then there's the really big sink, and that is the COS uptake by plants. So as you can see, those numbers here, they give sort of likely ranges. And you can see in very many cases, those likely ranges are really, really huge. So the COS budget is closed within uncertainties, but those uncertainties are really, really large. And by doing research on the uptake of COS by plants, we also contribute towards consolidating some of the uncertainties in the global budget of COS. Okay, so so this was more like in order to convey the conceptual idea of using COS as a tracer for GPP. So now we have to get down to the underlying mass, and there's two equations which we have to that end, and both are both apply at the leaf scale. So here we have the leaf scale flux of CO2, and according to Fick's law of diffusion, we have two endpoints here. So one is the ambient concentration of CO2, and this is the concentration of CO2 in the intercellular space. And we have one conductance, or we could also write it as a resistance, that is in between here. For carbonyl sulfide, we have a very similar equation, but here we've dropped the internal concentration of carbonyl sulfide in the leaf. So we are basically assuming, as stated on here, that it is close to zero based on a number of measurements. And then we have again the stomatal conductance, and we have an additional conductance that is termed an internal conductance here. And this is a bulk conductance. It combines the mesophyll conductance and enzymatic sort of conductances on the way. So now what we would like to do is we would like to estimate the CO2 flux here by using measurements of carbonyl sulfides. So how would we do that? Yeah, first we need to see which of these terms we can actually measure. And these are the red ones here, so we are likely to be able to measure the ambient concentrations of CO2 and COS. And of course, since we want to use the uptake of COS as a proxy for GPB, we need to be able to measure the uptake of COS. So these are known, but the other terms here are not known. So and we have way more unknowns than equations, and I guess all of you know that that's never a good thing. So fortunately, we still can get rid of a few of those, and the way we do that is by making use of the fact that when we know, for example, the stomatal conductance for COS, we can actually derive the stomatal conductance for CO2, knowing the ratio of the diffusivities. And in this case, it's we need to multiply the stomatal conductance for CS by a factor of 1.21, and then we get the stomatal conductance for CO2. So that sort of removes those two unknowns here, but we're still left with two more. And so what we do in this situation, and as very often in science, we come up with a parameterization. And the parameterization to get rid of those unknowns for CS is called the leaf relative uptake rate, in short LRU. And the LRU is the ratio of the deposition velocities of COS divided by the deposition velocity of CO2. So deposition velocity means simply the flux divided by the ambient concentration. This is an approach that's very popular, for example, in ozone research, because the higher the ambient concentration of ozone, the more vegetation will take up. And in order to compare different, for example, different places that have different ambient background concentrations, the deposition velocity is calculated. So it's a kind of a normalization of the flux with the ambient concentration. And the idea is now that when we have an estimate of LRU, we can sort of rearrange this equation here to yield the flux of CO2. So again, in red, the terms that we measure, provided that we know the leaf relative uptake rate, we can estimate here the CO2 flux, or I rather should say the GPB in this case. Okay, so let's suppose we can measure those terms here. So what value should we use for the leaf relative uptake rate? And this is a term that's actually been investigated for quite some time using leaf gas exchange measurements. And in a sort of big joint effort by the whole CRS community that was published in 2018 in Biogeosciences, I did a kind of a literature synthesis of published LRU values, and this is the distribution I came up with, and the median value is around 1.6. But you also can see that actually there's quite a bit of a spread, and if I'm not wrong, I think about 90% of the data fall into the range of 0.6 to up to 6. So there's quite a bit of a range, but overall the median value is 1.6. And initially it was this median value that was used, and the first one to sort of demonstrate how to use fluxes of CS to estimate GPB was a group from Israel, David Asaf and others, and they did the covariance flux measurements at a bunch of different ecosystems in Israel. And what they did is they would use the conventional FluxNet style flux partitioning, and that would be the black symbols here. And then they would use the flux partitioning with the equation we've just seen before, assuming the LAU to be 1.6. And as you can see, in most of the cases, the gray lines and symbols sort of overlap pretty nicely with the black symbols and lines, meaning that apparently this approach appears to work well, even though we've seen that actually the range in LAU is quite big. So when this paper came out in 2013, this was about the time that my group and I got into the COS business, and I was always a bit hesitant of using this universal kind of LAU value, and we'll see a bit later why that is the case. And so what we wanted to do is a different approach, and this is what I'll be showing in the following. So what we did is through a series of grants, we were able to come up with a multi-year kind of measurement approach. And what we did is in Europe, we would visit different eddy covariance flux towers with our instrument. You'll also see it in a second, and do concurrent CO2 and COS flux measurement at these sites. So for some of the sites, like this grassland over here, which is our own site, and also this pine forest here, which is our own site, measurements were going for 1 year, or at the pine forest site even for 2 years. For the other sites, we typically went just for a month and did a campaign. And in the following, I'll be showing results from the grassland, from a soybean field in northern Italy, from Mediterranean oak savanna in Spain, and from a temperate broadleaf mixed forest in Denmark. So at each of the sites, there was a flux tower, as I've said previously. This is our own site, the pine forest, Innsbruck, you can see over here in the back. And so each of the sites had an existing eddy covariance system, a sonic anemometer, infrared gas analyzer. And what we would add is our own analyzer that typically was placed in an instrument shelter somewhere at the base of the tower. And we would then suck down the air through a tube into that analyzer. And we would do the measurements at high frequency, so that we could actually then calculate eddy covariance fluxes of COS. So the instrument that we're using is a quantum cascade laser absorption spectrometer. So the company that produces it is in Boston, Aerodyne. And so this has a mid-infrared laser that, through a series of mirrors, is directed into a chamber that has mirrors on both sides, and then there are multiple reflections. So actually the path length is 76 meters, and the cell is about this size, half a liter of volume. And through that cell, we suck the air, and then we're getting those spectra. And with this instrument, what we're able to get is 4 species. So we're getting the carbon monoxide up here, water vapor. The strong line here is the CO2 line. And then this tiny guy up here is the COS. And with this laser spectrometer, basically the concentration of the gas of interest is proportional to this area. So basically it's the integration of this area. And yeah, so we can do measurements at 10 Hertz maximum. Very often, in order to improve the signal to noise ratio, we're measuring at 5 Hertz, maybe some cases 2 Hertz. But in any case, the measurements are fast enough to do eddy covariance. And since we have the CO2 here as well, you can also compare to the existing infrared gas analyzer measurements of CO2 and the fluxes that are calculated therefrom. Okay, so yeah, so in this sort of multi-site study, my PhD student Felix Spielmann, he came up with an approach where we actually would not need to provide an a priori estimate of the leaf relative uptake rate, but rather he and my group, we thought, why not treat the, so why not using CO2 and COS at the same time, sort of in order to harness the power of both those gases. So he came up with this idea to constrain the gross primary productivity by using joint measurements of CO2 and COS. And what underlies this idea is the sort of usual FluxNet style partitioning equations. So we have here a first term that represents the GPP. So in this case, this is the so-called daytime approach. So GPP here is assumed to be driven by radiation, by the photosynthetically active radiation. And then we have a ecosystem respiration term that's driven by temperature. Down here, we have the equation for the GPP, which we've seen before. And now the new thing is that we're parameterizing the LRU to be a function of the photosynthetically active radiation. And the form of this function implies that the LRU will take a constant value at high radiation. And then as the radiation decreases, it will slowly and then more quickly, actually exponentially, increase as the radiation decreases. And the reason for this behavior is very simple to explain. The carbonic anhydrase that catalyzes the COS is light independent, while photosynthesis obviously is not. So as the light decreases, photosynthesis goes down. So the plants close the stomata. So also the COS flux goes down, but it does not go down as quickly as the photosynthesis, and therefore the LRU increases at low light. And this is something that that this equation down here captures. And so what Felix would do is he would use a Bayesian inversion scheme, and he would have as measurements, he would have the net ecosystem CO2 exchange and the COS flux, all at the ecosystem scale, and then he would estimate all the parameters that you can see in here, and then he would compare sort of to the usual flux partitioning, just using the CO2. Yeah, so these are the results here shown as a light response, and the basic idea is that since light is central to this parameterization of the LRU here, and the GPP term here is kind of a obvious way to plot this. And what we have here in black in all the panels is the usual flux partitioning, just using the CO2, and what we call the flux partitioning plus, which is flux partitioning using CO2 and COS at the same time. And we would use equal weighting of the two fluxes. And so again, there's the 4 study sites, so the grassland, the mixed temperate forest, the cropland, and the savanna. And what you can see is that in all the cases, the black line is most of the time is below the colored line, meaning that with the flux partitioning that makes joint use of CO2 and COS, we're getting a higher GPP. Unfortunately, the difference, I mean, we were sort of, we would have been excited to see that this approach leads to a large difference. Unfortunately, it did not, but it's fairly consistent, and it's around in the range of 5% a larger GPB that we are getting with this joint use of CO2 and COS. We can look at the same data as at the online course and study the difference between the two GPBs. And this shown here, so always the colored line is the difference. So it's the conventional flux net partitioning just using the CO2 minus the new flux partitioning using both CO2 and COS. And you can see for most of the sites, it's actually, apart from morning and the evening, appears to be a constant kind of offset. Here at the soybean site, there's an interesting pattern, because actually around noontime there appears to be no difference between the two approaches, and there are differences in the late morning and in the later afternoon. And we believe this, for some reason, is an interaction between the radiation that the model takes into account and the vapor pressure deficit, which our approach does not sort of include. Okay. Yeah. So so this was sort of our idea at the ecosystem scale. So how to use COS without actually having to a priori make an estimate of the leaf relative uptake rate. But at the same time, and there's this actually quite recent work of mine, we've started sort of trying to better understand what may be causing the variability in LRU that is observed in these scale studies. And what we can do is we can take the equations that we've seen before, stick it into the equation of the LRU, do some rearrangement, and then we're getting this one. So now the LRU just depends on two ratios. So it depends on the ratio of the internal to ambient CO2 concentration, so the CI over CA ratio. And it depends on the ratio of two conductances, so the stomatal conductance for COS over the internal conductance of COS. And again, this factor 1.21 is just conversion of CO2 to COS stomatal conductances. And this equation here tells us in the first place that actually it's quite logical to expect variation in the leaf relative uptake rate, because we know from decades of research, for example, that there is quite predictable patterns in the CI over CA ratio across, for example, broad bioclimatic gradients from tundra temperate ecosystems to the desert, tropical forest, and so forth. And this has also been studied, for example, with isotopes of stable isotopes of CO2 in plant matter. So this is all very, very well known. And then there's also on a subdiurnal scale, there's variability in this ratio. And of course there's variability in the stomatal conductance. What we understand the least is this internal conductance to CS, because there are very, very few measurements. But taken together, this equation tells us that actually convergence to a value of 1.6, as it has been used initially, would be a kind of a coincidence, because it would mean that those two ratios would always, in a way, need to balance so that they would get this 1.6 here, which is unlikely to be the case. And so my idea was whether we could make use of a new approach in modeling gas exchange of plants, and that is optimality theory, to actually get a better handle on the LRU. And And what I'm using to design this is a model that's called the P model. It's been developed by Colin Prentice and colleagues in the UK since a seminal paper in 2013. And this model combines two sort of optimality theories. On one hand, the sort of least cost hypothesis, and this one is based on the idea that when plants take up CO2, there is a cost associated with that, because at the same time that they take up CO2, because they need to open the stomata, they lose water via transpiration, and that is sort of a cost. And actually, plants are thought to optimize the stomatal conductance so that the uptake of CO2 and the loss of water via transpiration is sort of optimal. And this is encapsulated here in this least cost hypothesis. And then there is what is called the coordination theory, and that idea is based on the fact that photosynthesis is either limited by light, or it is limited by Robisco and the concentration of CO2. And so these are two kind of separate processes, and plants need to invest into the underlying structures and enzymes and all that. And the idea of the coordination theory is that plants would invest into those two processes so that under the average environmental conditions, there's no overinvestment in either one or the other process, so that there is a co-limitation under average environmental conditions, and this is behind this coordination theory. Yeah, and together, and all that is sort of contained in the P model, and together the P model gives us everything that we need to do. We're getting this I over CA ratio, we're getting the stomatal conductance, we get it for water bit, but we can convert it to CS. And for the internal conductance, I've been using an approach that has been proposed in a previous paper. So we are scaling this to the maximum rate of carboxylation. And the idea behind that is simply that when plants have invested a lot in the carboxylation, it would make sense to have a relatively high internal conductance. Okay. And so I collated all the inputs, and actually this model here has just two calibrated parameters, and the inputs are temperature, CO2 concentration, VPD, photosynthetically active radiation, and yeah, so I'm having that on a global scale for the years 2001 to 2010 at 0.05 degrees resolution. So I can actually come up with global maps of the leaf relative uptake rate. And this slide here shows one of the results. So this is the growing period average leaf relative uptake rate, and the growing period here is defined as all months where the monthly average air temperature is above 0 degrees. And on the right-hand side here, you can see an average over all the longitudes. So now remember, we said in earlier works, it was assumed that the leaf relative uptake rate would be 1.6. So that's not even on the scale here. So this optimality model predicts leaf relative uptake rates that are way lower than what we've seen, what has been assumed in the past. They are still within the range. Remember, I said that the 90% range was 0.6 to 6. So they are, for most of the part, they are still in the range that has been observed. But to my knowledge, nobody's ever sort of proposed such low values of leaf relative uptake rate. And yeah, so the next step is actually to use those numbers in global inversion approaches, and in order to see what those low numbers sort of mean for the global budget of COS, because in the past, what has been found is that with a leaf relative uptake rate of 1.6 and accounting for the known anthropogenic and biomass burning emissions, the COS source in the ocean had to be cranked up quite a bit. And actually so far that it's actually not consistent with the measurements that we have for the ocean. So, and possibly the lower numbers that I'm calculating here might actually help to get the ocean source back into the range where the actual measurements tell us that it should be. Okay, so now to some of the complications, and one of the complications is that actually COS is not only taken up by the leaves, but it's also exchanged by the soil, and with exchanged I mean it can be either taken up or released from the soil. And what this slide shows you here is the ratio of the soil COS flux to the ecosystem scale flux, and we did this with the colors that you can see in the upper right corner. We will put a glass dome on top and actually do measurements of the soil COS exchange, and you can see that for many of the ecosystems actually it is around 5% or even lower. The only exception being here this Mediterranean savanna ecosystem, and we'll see in a minute why that is the case. So overall, even though soils sort of complicate the matter, because some of the flux that we are observing at the ecosystem scale may be due to the soil, it appears to be a relatively small contribution, at least when the leaf area index is reasonably high. Yeah, then we sort of also wanted to find out actually, so what is driving this soil COS exchange, and this was the PhD thesis of Florian Kitz. And in addition to the field measurements, what he did is he went into the lab and he incubated soil and did a number of tests with the soil samples that he derived from the field. So so he used untreated soil, so basically just sieved soil from the field, and he found that this soil was taking up COS and it was releasing CO2. So the CO2 flux here is in blue. So the units here are per gram soil. And then what he did is he would apply an inhibitor of the carbonic anhydrase. Remember, carbonic anhydrase is the enzyme that's in plants, and plants is responsible of taking up the COS. So he applied an inhibitor to the soil, and what he found is that when this inhibitor was applied, the COS uptake would sort of decrease. So this is actually what we had expected, because microbes, fungi, many of those have the carbonic anhydrase and are most likely behind this uptake that we're seeing over here. And if we inhibit the enzyme, then we are seeing a decrease of this uptake. So the COS uptake is not going to zero, and that means that there might be other processes that consume the COS, which are not dependent on the carbonic anhydrase, and also actually that is known from the literature. Then what he would do is he would apply glucose to the soil. So basically adding an abundant supply of readily available carbon to the soil, and as expected, adding this readily available carbon to the soil would increase the soil CO2 flux, interestingly, it flipped the sign of the COS. So now the soil started to emit COS. And then he did kind of a similar thing, and but this time he added litter into the soil, and again we saw that there was a huge increase in the soil CO2 flux, and again, at least initially, we observed also this flip to emissions of COS. However, there appeared to be some kind of decay in here, and mostly what we believe is going on is that actually over time the soils were incubated, sort of the pool of sulfur that could be volatilized here was sort of declining over time. And then at the end he sterilized some of the samples with gamma radiation. And so the idea was to kill all living stuff in the soil, and as expected, for most of the part, this really caused both the CO2 flux and the COS flux to become zero, indicating that it is really mostly of biogenic origin. And here I have to say that those measurements, they were made in the chart. Yeah, so this was kind of meant to be an analysis. So what's going on in the soil, and also in the field, we would sort of explore different situations in the field in order to find out what the variability is. And for example, here in the savanna ecosystem, we will have those soil rings placed either in the open spaces where there's just grasses, in the interspace between trees, and then below the trees. And for the measurements that were made in the night, so these dark bars here, this was not a big issue, so no big differences. But when measurements were made during daytime, then in the open spaces and in those interspaces, we would see large emissions of COS, and this appears to be an abiotic process. So when UV radiation hits, for example, litter on the soil, it can sort of cast out COS molecules from that plant litter, and this is then something that we are picking up with our measurements. Yeah, really recent work by Florian, who's in the meantime finished his PhD, is still in the lab, and here in the lab he's trying to disentangle the role that roots are playing in the soil COS exchange. And to this end, he's having those pots, and some of the pots have plants in there, like 1, 2, 3 over here, and the other pots just have the soil. And he's comparing the soil COS exchange between those pots. And so these are examples for beech. And apart from measurements in February, when the peach had no leaves, actually, what we can see is all other times, whenever there's roots present in the soil COS emission is smaller compared to soil only, which means that either the presence of roots in a way inhibits the emission of COS, or there's actually an uptake of COS associated with roots, either directly the roots or the microorganisms that live in the rhizosphere. Yes, and finally, Florian has also been looking into whether we can see COS fluxes from litter, and this is just a few examples. And the fluxes appear to be small, but there appear to be also differences. We can have clear emissions, but also occasions when there is actually uptake of COS by the litter. And all that sort of complicates the use of COS at the ecosystem scale a bit. But so far, what we've seen is that mostly those other fluxes appear to be small compared to the strong uptake of COS by the leaves. So with that, I'm almost done. So in conclusions, we just had, I think 2 weeks ago, we just had the 3rd international workshop on COS, and during the past 10 to 15 years, this small group has been really very, very active, but still, I realized that there's really a lot of things we yet do not understand very well. The variability of the leaf uptake rate is one of those issues, and since this is really key to using COS as a proxy for GPP, it's something that limits our understanding, and we need to really make progress here in order to better use COS as a proxy for GPP. What I've proposed today, using this optimality theory, is a completely new way to look at the leaf relative uptake rate. The numbers that I'm obtaining this way are very, very low, and we need to understand whether this is useless, it may be, or whether possibly it actually helps to reconcile the different components in the global COS budget in a way that actually makes sense. So it's really too early to say. I've been showing you that the soils may either be a source or a sink, but typically, at least when the leaf area index is reasonably high, then the soil actually is something not very much to worry about. But this may be, of course, different if a field, for example, an agricultural field is fallow, so there's no vegetation or something like that. Yeah, so the next approach, or the next step in my view, is to continue this idea that we did in the PhD thesis of Felix Spielmann, using multiple constraints. So he used COS and CO2 as joint constraints, and the next logical step is to do that same thing with another really promising candidate, and that is the sun-induced chlorophyll fluorescence. And actually, this is something which we've already sort of exploited. These are measurements taken in a Mediterranean pine forest in Israel during a heat wave. So this is about 10 days. And here in gray, you see the air temperature, and you can see how the air temperature ramps up. And then there's a storm, and it goes back sort of where we started. So this is quite typically in this area in spring. So when a certain constellation of pressure differences causes air from the desert to flow to the site, and then we're seeing these heat waves. And what we can see here, the GPP during this heat wave, it really declines dramatically. Although once the temperature is back to normal, also the GPP recovers quite quickly. But this made us sort of curious to understand this decline in the GPP. What does it mean for COS? And what I'm showing here for COS is the ecosystem relative uptake rate. So this is simply the deposition velocity of CO2 divided by the deposition velocity, the deposition velocity of COS divided by the deposition velocity of CO2, but not using the GPP, but really the net ecosystem exchange. And what you can see here is during much of the heat wave, actually the ecosystem relative uptake rate was quite constant, but then at the peak of the heat wave, we saw this strong increase. And what this means is that this uptake of CO2 actually decreased much more than did the uptake of COS. So what what certainly happened, and this can be seen also in the latent heat flux, is the plants closed the stomata in order to prevent transpiration. And this, in a way, is responsible for this relative constant ecosystem relative uptake rate, because the stomatal conductance affects both the uptake of COS and the uptake of CO2. So this very strong increase here, what we believe is happening, that here we are seeing non-stomatal limitation of photosynthesis really at the peak of the heat wave. And this is corroborated by the measurements of the sun-induced chlorophyll fluorescence. Here we have the red and the far red chlorophyll fluorescence. And we can see that for much of the heat wave, this was pretty constant. And then we saw at the peak of those 2 days, we saw this drop in the chlorophyll fluorescence. And we also believe that this is an indication of severe sort of physiological stress. So this at least is sort of gives us some flavor of how we could use another constraint, so in this case chlorophyll fluorescence, in order to diagnose what is going on in the ecosystem. Yeah, and finally I would like to mention, although I haven't talked about it at all, that because the stomatal conductance is sort of a key regulator of the COS uptake, COS fluxes are also linked to transpiration, and actually can also be used to do another form of partitioning for the water fluxes, which is something that came up during the past couple of years as the new flux partitioning the evapotranspiration into transpiration and evaporation. And also here, COS can make a contribution. Okay, so with that, I'm actually done and would be happy to take your questions.
If you have any questions, you can just walk up to the mic and ask those.
Georg, I have a couple questions.
Go ahead.
What was your methodology for estimating conductances at the canopy scale? And then secondly, are there any significant differences with respect to a C3 versus C4 photosynthetic pathway with some of the COS measurement?
Yeah, good point. I forgot to say that. The C3 and the C4 pathway are dramatically different in terms of the leaf relative uptake rate. So C4 species have a much lower leaf relative uptake rate, and so one explanation for that is simply because the CI/CA ratio in C4 plants is typically 0.4, 0.5-ish, while for C3 plants is more like 0.7. And that already explains much of the difference in the leaf relative uptake rate. Also, the optimality model that I've been using is actually strictly for C3 species. Yeah, so having said that, I think there are very, very few measurements on C4 species, either like leaf scale. There are a few, but I don't know about any ecosystem scale measurements. So regarding the first question about conductances and the approach that we took, so actually I wasn't showing any conductances, like field measured conductances here. It is though something that we are doing. So basically, this is something that Felix is working on currently. So he's disentangling the total conductance into the aerodynamic part, the boundary layer part, the stomatal part, and the internal part. And to that end, he uses like it's often done the water vapor flux measurements, and then inferring the internal conductance as sort of the remainder. Yeah.
Thank you.
Hi, I had a quick question. Can you hear me?
Yes, I can.
I was just curious to get your. My name's Dylan Millay. I'm in the department. I enjoyed your talk. I was curious to get your perspective on, what's our status right now in terms of the sort of observing network for COS. Are we sort of to what degree are we limited by measurement coverage?
Yeah, so I think we're in a situation like we have been for CO2 30 or 40 years ago, when people started doing eddy covariance flux measurements. So it's really like at the CS workshop 2 weeks ago, we were about 30 people, and maybe there's 10 to 20 more that weren't able to attend. But really, that is about the size of on the international scale, the size of the folks working on this. And but that is distributed between like leaf scale people working like in the lab, ecosystem people. Then there's a whole bunch of modelers that do inversion studies or do regional scale studies. So really, we're limited by the size of the group, and there's only so much work that we can get done, and we are also severely limited by measurements of all sorts of things, like leaf gas exchange. There's maybe, I think we haven't measured more than 20 different plant species. And probably for ecosystem scale flux measurements, we also haven't measured at more than, I think, 15 or 20 sites. And at some sites, it's just a month. There's, I think, only 2 sites where there are long-term measurements that go beyond 1 year. So we haven't really touched upon interannual variability at all. Then some of the concentration measurement stations from NOAA and other organizations, they have COS being measured with high precision. There's now satellites that sort of allow estimating COS, like integrated columns. But yeah, we would really need more data and more people working on this topic.
Sounds like good job security for the future.
Yeah.
Hi, I have a question. Can you hear me?
Yes, I can hear you.
Yeah, I want to know, how did you get the global map for the IU? Did you use a P model?
Yeah, so I used the P model and the corresponding drivers, and so this is temperature, VPD, photosynthetically active radiation, CO2.
Okay, so
and pressure.
yeah.
And it has just 2 parameters. I mean, I have to say I'm a bit of a fan of the P model because it just has 2 parameters and they are already calibrated. So basically, it's really very easy to run on a global scale. Having said this, and like the authors of the P model, they have also gone a long way in validating it. So at the leaf scale, also at the ecosystem scale. So what I don't know yet really is those low numbers of the LIU we are getting, whether that is of really any value. So we really need to see.
Yeah, I'm very interested in the P model. So with the photosynthesis optimal framework, so can you distinguish the specific controls from the environmental factors such as the PAR, VPD, temperature, like that? Can you distinguish those different impact?
Yeah, I mean the P model is like the core is the Farquhar von Camera Berry standard model, which I would say is state of the art. So all you can do with this model, you can do with the P model.
Okay, so with the padding, the global padding, I say the IOIU is most higher in the tropics, but the lower in the high latitudes. So in your opinion, which factors control those different? It doesn't seem the PR, the temperature like that.
Yeah, so this is mostly temperature and VPD. It's not so much the radiation, it's mostly temperature and VPD, because the map I was showing is a growing period average. So for the high northern latitudes, this may just be the average over, I don't know, June, July, August or something like that. While for the tropics, it may be a year, or most likely it's a year-round sort of average. Yeah, so the higher LAUs in the tropical regions is mostly temperature and VPD driven.
Okay, yeah, in your GRL paper, you built a nice functions with the PR. So did you consider, I mean, the functions about the VPD, the temperature like that?
Okay, yeah, that's something I haven't actually thought about. So the point is that in the geophysical research letter papers, so this was based on 4 study sites. And there were differences between the sites, not so much in the slope,
yeah,
kind of thing, but more in the value at high radiation. So so here we are again back at the issue that we have not global coverage of, we haven't yet covered all major biomes. So actually, I think we have no published study from the tropics of COS fluxes. So using this function that we used in there would be quite a bit of extrapolation, I think, at the moment. But we could, for selected biomes, we could try to actually compare it. There's one issue here, though, and this is that there's also scaling involved when we go from the leaf to the ecosystem scale. Because in a plant canopy, so the radiation decreases about exponentially with depth. So that means that the leaves, the uppermost leaves, they will have a relatively high LAU, a relatively, sorry, other way around, a relatively low LAU. But as we go deeper in the canopy, because the radiation decreases, the LAU goes up. So actually, when we do flux measurements, we integrate over all that, and we're getting an ecosystem scale leaf relative uptake rate that is like weighted average of this. So when we want to translate between leaf and ecosystem scale LRUs, we need to do those scaling. And the same holds for the VPD, which is also different between the uppermost leaves and down in the canopy. And in the leaf scale LRUs that have been measured in the leaf chamber, they usually are measured in a situation where the boundary layer conductance is close to infinite, because we steer the air very much. And so the boundary layer conductance typically plays no role, while in nature, this may not necessarily be the case. So at low wind speeds, the boundary layer conductance may play a role, and it is more likely to play a larger role, again, deeper in the canopy. So what I'm saying is, yeah, we need to be aware of the scaling issue between leaf and ecosystem LAUs.
Yeah, very good work. Thank you.
You're welcome. Okay. So have I answered all the questions in the chat? I think so.
I think so. I think we're ready to wrap up.
Okay. All right, thank you, Georg. Thanks very much for staying up to 11:30 at your place there. So appreciate your time, and we'll be chatting to you soon. Thank you.
Yeah. Thanks and bye.